Hi everyone, I'm Fiona. I'm from the National Centre of Biotechnology Education at the University of Reading. Thank you for joining me for our first video all about proteins. So let us start at the beginning. I hope you're all sitting comfortably. Our genes are lengths of DNA and they encode for all of the proteins that we make in our bodies. So this is achieved by DNA first being transcribed into RNA, and that is then translated into amino acids which come together to form a protein. A good way then to remember the order of events is that you can't translate something unless it has already been transcribed or copied. So the DNA that we have in our nuclei is far too big to make the journey through the cytoplasm over to the ribosomes to be translated. So it first needs to be transcribed, so it needs to be copied into RNA, which is then much smaller lengths. So it can come out through the cytoplasm, out of the nucleus, and then go and be translated on a ribosome. So therefore, the important sequence of events to remember then is that the DNA first has to be transcribed into the RNA, and the RNA then is translated into the amino acids and therefore the protein sequence. So in our bodies then, when a particular protein is needed, so this will be in a particular area of our body, the cells in that area, so sometimes it can just be a single cell, sometimes it will be a big group of cells, will start to go through the process of making that new protein that is needed. So our RNA polymerase will bind to the gene which is specific for that protein that is needed. It will bind to a particular sequence. Now, this is known as the promoter, and this tells RNA polymerase exactly where that gene begins. So it will bind onto there and it will then unzip the DNA, and this is in a very localized region just specific for that gene. So it'll unzip the DNA, because obviously we know DNA is double-stranded, so it'll unzip it, as you can see here, and this will form a little bubble. So the RNA polymerase will then start to move along the antisense strand of DNA, and as it goes along, ribonucleotides will be added, and these will be complementary to this DNA strand that it is moving along. So the RNA strand will grow in a 5-3 direction, and the resulting RNA strand will then resemble the other DNA strand, so the sense DNA strand. And obviously the only difference then will be that it has uracils instead of thymines. So transcription then stops once the RNA polymerase gets to a specific sequence again. So like the promoter sequence, which told the RNA polymerase where to start, this is called a terminator sequence, and it tells the RNA polymerase that is the end of the gene. So it will stop transcribing when it gets to that point, and the RNA will then be released. Now, in eukaryotes, at this point, the RNA is called pre-messenger RNA, and that is because it still has its intronic regions, which need to be spliced out. So we just want the exons, which are the coding regions, to then be translated into the proteins. So these regions, the intronic regions, are gotten rid of. And then also the RNA needs some protection before it leaves the nucleus and goes out into the cytoplasm, because otherwise it will just be degraded by RNases that are present. So it has what is called a 5' end protective cap put on, and it also has what we call a poly A tail put onto its 3' end, and these are just some adenines that are added on. Now, both of these then will help protect it on its journey to the ribosome. So once the messenger RNA has left the nucleus and it's found its way to a ribosome, it can then be translated into its amino acid sequence. Okay, so how then is RNA translated into that important amino acid sequence and then into its fully functional protein? Well, it obviously starts with the sequence that has been transcribed from the DNA into RNA, and how that then is read, how it's translated by that ribosome. So the ribosome reads 3 bases at a time. So these are called codons, and they don't overlap with each other. It's not like a sliding thing where it just picks 3 bases at random. So it will start with what we call the start codon. So in eukaryotes, this is AUG, and it encodes for the amino acid methionine. And it will then, so it will start here, and then it will run along the length of RNA, taking each 3 bases as it finds it. Now, as I said, it isn't overlapping the code, but it is degenerate. So most amino acids have more than one triplet sequence that codes for it. So for an example, you can see here the 2 codons which are highlighted in purple both code for leucine. And once the messenger RNA reaches the ribosome and the ribosome complex is formed, so this is made up of a small ribosomal unit and a large ribosomal unit. So once they've come together, starting to move along the RNA and translate this into amino acids. So this is done by transfer RNAs. So as you can see here in my diagram. Now these bring along the amino acid which is coded for a particular codon, and they have on their bottom an anticodon. So this is complementary to the RNA codon. The amino acid that it has attached to it will be specific for that codon. So the transfer RNA then docks onto the RNA strand and will wait there. If it's the first one, it will wait there then for the next transfer RNA to dock next to it. And a peptide bond will then form between the 2 amino acids. So this is obviously a condensation reaction because a peptide bond has been formed. So the first transfer RNA will then detach, and that will then go off back into the cytoplasm, and the ribosome will move along to the next codon, and the next transfer RNA will dock. And the same thing will then happen. A peptide bond will form then between that 3rd amino acid and the 2nd amino acid. Now this will continue until it reaches a stop codon, and that at which point the translation will cease and the new polypeptide will then be released. So the structure of a protein is firstly down to its primary structure. So since the properties of amino acids present in a polypeptide chain will affect how it folds and what sort of properties the protein will have. So there are 20 amino acids in total, and the simple structure of them all is the same. So they have an amine group on the left, as you can see here, a carboxyl group on the right, and then a side chain which is shown here as R. So this varies then the side chain between different amino acids. So for example, methionine and glycine are shown here. Glycine is very simple, with just a hydrogen as its R group. Glycine is both polar and hydrophilic in its properties, whilst methionine, as you can see here, has a much longer R chain, and this makes it much more hydrophobic, and therefore you would generally find this buried in the middle of a protein structure. Methionine, interestingly, is also one of only 2 amino acids which is sulfur containing. Okay, so to recap then, today we have been through how DNA is transcribed into RNA, how that RNA is then translated into amino acids. The amino acids then go on to form a polypeptide chain, which are connected by peptide bonds. And I'm going to just leave it here then for this video. And you can all go and have a little break. And then hopefully join me again for my next video, in which I will be looking at how proteins fold into their secondary structures, and then go on to form their tertiary and quaternary structures, and then how this relates to their function. Additionally, I will also be telling you a little bit about what happens when things go wrong. So thank you very much for joining me for this video, and I hopefully see you again soon.